I would like to give you a brief introduction to the webinar, so I'll just start sharing my screen with the first slide here. So first of all, thank you all for joining us this evening on this pulmonary fibrosis webinar on translational science and pulmonary fibrosis from patient to cells and back, increasing success in drug development. We are happy to have 3 excellent speakers here who will share their insights in pulmonary fibrosis. As always, we hope so. I'll just take the next slide here so you can see the program. It's here. So this is the program for tonight. And we are still waiting for Naftali, so we will just give the word to Line to begin with, but otherwise we will keep the scheduling. Then, as always, we will like this to be a very engaging session, so please don't hesitate to ask questions through the private chat. We will be answering the questions during our dedicated Q&A session and at the end of the webinar. So please help making this an engaging and insightful experience for everyone. So with that, I will just like to say welcome to Line Erol, a very skilled data scientist with a strong focus on applied machine learning. I'm super lucky to work closely with Line, so therefore I also know that while she works with sophisticated machine learning techniques, she has a remarkable ability to create beautiful and intuitive slides and make even the most complex concepts easy to understand. In her presentation, Leverage Machine Learning for Patient Stratification and Advanced Analytics, a CPD case study, Line will demonstrate how cutting-edge using machine learning can be used to identify patient subgroups, optimize clinical decision-making, and enhance our understanding of complex pulmonary diseases. Please welcome Line Erol.
Thank you so much, Sarah. I will just share my screen. Yes, can you see my slides? I think you can. Again, thank you, Sarah, for introducing me, and thank you for letting me speak today. And as Sarah said, I will talk about how we use machine learning for patient stratification. And I'm going to come up with some working examples, both on supervised machine learning and also unsupervised machine learning. I'm mainly going to talk in the lung disease known as COPD, but I will also quickly deviate over to IPF as well. Yeah. So first, what is COPD? Well, COPD is the 3rd leading cause of death worldwide, and in 2019, it resulted in over 3 million deaths. And if we start ranking the fibrotic diseases, it's actually the 2nd leading cause of death. So when it comes to COPD, it is very prevalent. When it comes to the age group from 30 to 80-year-old, we actually see that 10% of the world population have this. Of course, there's some regional differences, but it is a worldwide problem, and it's only going to increase in the future with more pollution. Then this figure is all wrong. I was about to show that this disease is mainly known as the smoker's disease. So smoking is the primary cause of COPD, but there's of course other things that add up in this horrible equation. And unfortunately, there's still no. These slides are really there. Unfortunately, there's still no cure to this. When the lung damage has occurred, we are not able to restore the damaged tissue. There is medication out there to stop progression and relieve symptoms. For example, this dupilumab that is shown to increase lung function. However, there's still a need for better medication and also a need for biomarkers, since some medication types only work for a subset of patients. So what is COPD? Well, COPD is characterized by airflow limitation, and this airflow limitation results from various mechanisms and includes both loss and gain of tissue. And the clinical phenotype have often been described what part of the lung was affected. So if it was a persistent injury to the small airway lumen, we would see an increased release of mucus and a thickening of the airway wall. Where on the other hand, if it was down in the smaller alveolar space, then we saw a destruction of attachment and loss of elastic recoil. We can take an even closer look at this, and we can look at how does it actually look when we look at the extracellular matrix. And here we also see that there are some changes, some dramatic changes to the ECM. On top here, we see how a healthy lung looks, and on the bottom, we see how these different processes happens and looks during disease progression. For example, if we go back to the what's it called, the small airway lumen, we do see that what's it called? We can see that these endothelial cells activate fibroblasts, which then starts to producing a lot of collagen and ECM fragments, which is then deposited in the tissue and making it thicken. On the other hand, if we look at the alveolar space, we see a different process. Here we do see how these inflammatory cells start to release proteases that then degrades the ECM process. So in COPD, we both have this formation and we have this degradation, and in both processes, the cleaved ECM fragments can actually be detected in the blood. And now I'm going to take you on a journey back to the 1960s, how we looked at patients back in the days. So here's a painting that represents how patients could look. When you came into the doctor, you could look either like a blue bloater, you would be a heavy person, and you would do a lot of coughing, or you would be a pink puffer, where you would be very, very skinny, and you would have a really hard time breathing. But we know now that this is not true. We know that COPD is a disease with a heterogeneous manifestation, and patients can have signs of both these phenotypes, but also others. So we know that it's not just one disease. We claimed that back in the 1960s, and we're only getting better at it. But when we look at these phenotypes, we also see that, well, phenotypes are pretty exclusive. Like, that's one patient, this is how it looks. But we know that most patients can actually present one or more molecular mechanism underneath. So that was why in 2016, this precision medicine strategy was launched that was called endotypes. And it supports the idea of these different molecular mechanisms that can coexist in the same patient. And the goal with endotypes is simply to tailor a treatment to the underlying molecular mechanism and treat the underlying mechanism instead of the symptoms. And then the big question is, of course, how do we find these endotypes? And how many are there, and do they actually exist? And here we hope with this ECM technology I presented earlier, that we can use some of these different biomarkers that represent different processes, kind of like a panel, so we can improve not only our understanding of disease, but also identify these endotypes. And we have seen multiple times that these ECM biomarkers works in COPD. This is some work made by a few of my colleagues, where they showed that increased elastin degradation in the blood is associated with a higher mortality in these patients. Here's another study where we, again, some colleagues of mine that showed that collagen turnover is related to annual change in the lung function. So we know that these biomarkers work very well one by one. But today I'm going to present what happens when we try to combine them. So that's what I'm going to cover. This was the introduction. Now I'm going to go to the more working examples. The study I'm working with, or we are working with and have in-house, is this cohort called Eclipse. And Eclipse is a retrospective cohort with over 2,000 COPD patients from 12 different countries that was followed for 3 years, where there was collected a lot of multifaceted data, which covered both proteomics, genetics, lung function, CT scans. We have a lot of information on these patients, and this is what you need when you have to do advanced analytics and machine learning. So that leads me to the overall aim of this project, which is basically, again, taking this cohort, matching up with these ECM biomarkers, and then use sophisticated methods. Here I'm talking about, of course, the big buzzword, machine learning. And today I'm going to cover both supervised machine learning, I'm going to cover unsupervised machine learning, and I'm going to explain about this explainable AI concept. So I hope you stay with me for this. So first I'm going to cover what is this supervised machine learning and how do we use it for endotype discovering. And the thing with supervised machine learning, hence the name, is that we need to know the outcome up front. So here we actually take a known phenotype. We could look at, for example, the phenotype emphysema, and then try to identify these molecular mechanisms behind the phenotype. Like, what is it that actually drives this known phenotype? And I'm going to explain it both on a global level, but also on the individual level page. So here's just a quick overview of the study design. We took these 2,000 patients from the ECLIPSE cohort, then we filtered them quite a lot, and then we looked at the CT scan and divided the patients into outcomes. So did they have emphysema or did they didn't have emphysema, and how severe was the emphysema? For today's talk, I'm only going to focus on the no emphysema group and the mild emphysema group because they're very, very close in biology. And this is where machine learning actually can do us a lot of good, because there's a lot of complexity in determining this. So this is our input to our outcome of the model. For our input, we included 7 different biomarkers covering 4 different processes. So we both have some inflammation, we have some elastin breakdown, we have different degradation and formation of intestinal matrix and basement membrane, and then we further included 151 other features as well, which you can quite easily measure in a clinic. For example, questionnaire, exercise capacity, your pulmonary function. We then took all of this and we included it into something called an extreme gradient boosting model. And this XGBoost model is actually just a supervised machine learning. And you can pick whatever model you want. You can pick a neural network, you can pick other much simpler decision trees. But what I like about this model is that it is a lot of decision trees. You just run computational fast on top of each other and you try to minimize an error. When you have the result of that model, we put it into an explainable AI framework. Here we use something called SHAP, Sharply Adaptive Explanation, where we kind of reverse engineer. So you take the result from this model, and then you try to determine, well, what actually make the model predict what is predicting? Why is it saying that this patient has emphysema and this patient don't have emphysema? So first you need to evaluate such a model. And here I'm showing an area under the ROC curve. I think most of you might know it. Otherwise, I would say that a score of 0.5 is a random model. A score of 1 is a perfect model. So when you see a score of 0.7, you're like, that's maybe not so impressive. But when you're working with very, very complex biological question, 0.7 is actually pretty standard. Even more important, we can go in and look at specificity and sensitivity. So we can look at what does the model actually do. Well, the model is really, really good at detecting people that has emphysema, not so good at detecting patients that don't have emphysema. So it's a very nice screening tool. But you can, of course, adjust the threshold depending on what your application is. You can do some cost balance if you only want the more predictive patient to be involved, and then increase the specificity and lower the sensitivity. But that's up to the application. More importantly, I want to talk about this explainable AI, because one thing is a model can tell you a yes or no question, but the big question is also, why is it saying this? This is what of interest to a clinician or a patient. So again, we're using this SHAP to kind of reverse engineer on a global level. So here's a SHAP plot, and here I show top 8 of the 158 features we put into a model. Each dot on each line represents a patient and how that feature impact the model decision for that patient. And we can here see that the top predictor is a very well lung function, which is an FEV1 FVC ratio. So to explain it a bit better, what the model shows us on the global level is that you have lower of this measure, meaning worse, then you will probably have emphysema. On the other hand, if you have a high value, a good value, then you probably don't have emphysema. Pretty simple. But we also have to remember that this is one big puzzle when you work with machine learning. The model looks not only at this one feature, it looks at all the 158 features and tries to predict based on a combination of all of those features. So in our top 8, it's also very interesting that we see 3 biomarkers representing both formation, PRO-C6, degradation, C3M, and also elastin breakdown. And again, it's important to remember that a patient does need to have all 3 elevated to have emphysema, but rather see it as 3 potential endotypes that combines shows this phenotype. Again, on a global level, if we look at PRO-C6, we can see that there's actually not really a difference between the no emphysema and the mild emphysema patients. So that means we have to go even deeper. We have to go on to a patient level and see how is this actually reflected in a patient. And again, we can use explainable AI. This is just a made-up example. I'm not going to show individual patient data. So let's say a patient goes into the clinic and the model say, well, you have a 38% risk of emphysema. Well, that's really nice. But I think most doctors and most patients want to know, why is this and what can I maybe do about it? And here you can ask the question again, well, why is that? And here you can see for each individual patient why the model determines what is determined. So by utilizing this explainable AI on individuals, we can potentially find, well, underlying endotypes. Some of them would be found in multiple patients. Some of them will maybe only found in a handful of patients. And hopefully this can be used in future trial design and to get the right drug to the right patient at the right time. So that was one use case I wanted to discuss. Then I want to discuss the other one, and I think most people know about this one, and that is unsupervised machine learning, also known as clustering. So clustering is different because you're not telling the model what it's looking for. You're simply saying, I have some data and I want a pattern, and hopefully that's an endotype. So we tried that. The next case I'm going to show is going to be another lung disease called IPF. And it's simply because we're still working on the COPD model. We're using the same methodology, but it's still undeveloped. So IPF is also a progressive lung disease where the clinical trajectories varies a lot between the patients. In this study, we did this as kind of a proof of concept. We included 100 patients from 3 Danish referral centers, and then we measured 10 blood-based ECM biomarkers that covered these different processes I talked about. And that was the only one we included in the model. We then performed something called principal component analysis, where you take these 10 dimensions of data, you try to squeeze it in a bit to remove some of the noise, and then we did clustering on top of this. In this case, we used something called k-means clustering, and we tried it to determine how many clusters there might be, and is any of those prognostic. And to give the result, we found clusters. You always find clusters when you use unsupervised machine learning. The question is, does they actually mean something? And we found these 3 clusters, and when we looked at them, we had kind of a red cluster, a blue cluster, and a green clusters. And we started to look at the endotypic differences across biomarkers shown here on a spider plot, where we clearly can see that the patient have clusters into this blue group, which is the low biomarker group. We have the green group, which is a mid-level group, and we have a red group which have high level of most of the biomarkers. And we see that there's approximately 6 biomarkers that segregates the patient, and these biomarkers span 3 different processes: ECM remodeling, neutrophil activity, and degradation. So these findings kind of suggest that monitoring of these 3 processes could be sufficient to identify IPF phenotypes. If we then go back and take a prognostic view, because again, you can always find clusters, but what does that actually mean? We see that the patient, here we see the patient decline over a 3-year period, and we see that patients with low biomarkers values at baseline are stable until around year 2, and then with a slow decline. Whereas patients from cluster mid and cluster high show a steadier decline with a significant decline in cluster high at year 2. We can also look at mortality, but mortality didn't show as good as a result. But we did see that when we looked at the risk ratio between the low cluster and the high cluster across all time points, that there was an increase in mortality starting around year 1, and then the trend kind of continued. So you can actually use these prognostic. But another thing that's important to remember when we work with a lot of, especially biological data, is that there's always a connection between things. So we had to look at the baseline demographics as well to determine whether these biomarkers do have prognostic relevance or whether biomarkers are simply a proxy for something else going on in the body. So on the screen, we see the patient demographic for all three clusters. And if we look to the p-value column, we can see there's not really a difference between the groups. And that indicates that these ECM biomarkers on their own can predict disease progression better than any clinical parameter can. And this was a proof of concept study we did in 2003. And just a few months back, this very, very nice paper from Feinberg et al. Was published in the Lancet Respiratory Medicine, where he used a larger IPF cohort. I think he had 2 cohorts even, and measured 13 other blood-based biomarkers. And in this paper, they also identified 3 clusters with different prognostic, highlighting the prognostic potential of these endotypes. And finding like these could hopefully improve patient selection for future trials by including the right patient, or even if you're testing a specific drug, then you get the right patient with that molecular mechanism being disrupted. And you need that to have a drug working. So I think my time is running out. So I'm just gonna go to the conclusion real quick, and highlighting that machine learning combined with the biomarkers, in this case ECM biomarkers, offers us a real world insight into disease progression. And it could improve our patient selection and could then improve clinical trial design. Furthermore, I didn't talk this much about it today, but you can also use a combination of this as a translational tool for biomarker and target discovery. And of course, as I mentioned on the last slides, you can identify prognostic endotypes. So with that, I'm just going to end and acknowledge a lot of people. It takes a lot to make stuff like this. And again, looking forward to the Q&A. Otherwise, if you have any question, please hit me up on my email, and I'm happy to discuss. Thank you.
Thank you so much, Line, for that talk. And I can see that there is questions coming in the chat, but for the interest of time, I think we will have the question in the Q&A at the end of the session. So I would just like to give a very warm welcome to Dr. Naftali Kaminsky that has joined now. He's a very well-known key opinion leader in pulmonary research. Dr. Kaminsky is Boehringer Ingelheim Endowed Professor of Internal Medicine and a Chief of Pulmonary Critical Care and Sleep Medicine at Yale School of Medicine. And today he has this very interesting topic, investigating fibrosis mechanism, what drives fibrosis in lung. So his pioneering research combines genomics, system biology, and clinical science to uncover underlying mechanisms of fibrosis and drive innovation and therapeutic development. So please welcome Dr. Kaminsky.
Thank you. Can you hear me?
Yes, we can hear you.
And let me share my screen in a sec. So it will come up in a sec. Okay. Can you see my presentation mode?
Yes, we can see it.
Perfect.
Thank you.
Thank you. So really excited to be invited to give this talk today. I'll try to sort of speak about our work mostly on single cell, a re-understanding of pulmonary fibrosis and what are the key cells. The people that contributed to this work, and I'll mention it up front, is Taylor Adams and Jonas Schupp and Aurélien Justet and Nabala Boussen and Ivan Rosas and John McDonough, and Zitin Yang, and many others. And again, the way the disease that I study is basically progressive pulmonary fibrosis and IPF. This is a heterogeneous group of conditions that lead to progressive lung scarring, probably around 400 or 500,000 people just in the US. Only cure is lung transplant. Many causes: genetics, environmental exposure, autoimmune, and idiopathic, which is IPF. And usually the disease is most lethal when it is associated with a very, very stereotypic pattern, which is this progression of the disease from the periphery into the center with honeycomb cyst, and with this lesion of ethology, the sort of what we call fibroblast focus with normal lung adjacent to it, and you see it here, and a layer of epithelial cells. And we do have the scientific community, and part of it is my work, of course. We have developed an approach in which we can basically show you the concepts of the lung. So this is again the IPF lung, and you can see my laser printer. And our hypothesis is that what's happening in IPF is there are recurring microinjuries that then cause epithelial cell injury, potentially activation of cells that don't die, matrix remodeling, and then cell proliferation and migration. And if you have a genetic predisposition, exposure, telomere attrition, or aging, you will develop this lesion. So this is a really cool GIF, and I'm sure I'm very proud of it. But does it really explain what happened? So the truth is it does not. This model does not explain the cellular repertoire of the lung. The lung is rich with multiple cells, doesn't explain the anatomic features. It does not explain relentless progression. Most scars do not progress. And so it's not sufficient. So luckily for us, to understand the tissue, we need to understand the cell types and the organizational roles in the tissue. And in a very simple way, in the human lung, we have the airways and we have the alveoli. Airways sort of bring air, so they have a lot of cells to do sort of innate immune protection and bringing the cells. And then we have sort of the alveolus, which was evolved for gas exchange, so very, very thin structure. And in it, you have the alveolar type 1 that does gas exchange, and a lot of other cells that sort of support the process. So the primary cell in the lung is the alveolar type 1, and potentially the aerocytes, which is the endothelial cells that's adjacent to it, that they do the gas exchange. And many of the other cells that many of us, and of course myself, have developed their careers to understand, like alveolar macrophages, general capillary cells, alveolar type 2 cells, or alveolar fibroblasts, they're all sort of support cells. So it would be interesting and important to understand how this is affected in the lung. And around 2000, we published and released the IPF Cell Atlas paper in Science Advances, and a website that contained also data from other papers from Nick Banovich and other John Kropsky, and released the Cell Atlas. It was sort of a difficult time for humanity. People were sitting in the rooms because of the pandemic. It was good for website. So indeed, we got almost 15,000 unique users from 56 countries, 5 continents, that search for multiple genes. And again, you can scan for it here and access it. You can see at the time, of course, ACE2 was sort of the top. But what did we learn with a single cell and single nuclear sequencing in IPF? So we analyzed numerous lungs, extracted either cells or nuclei, and then isolated. So the first thing we saw was actually very obvious. There was a loss of alveolar epithelial cells. And ciliated cells, the airway cells, showed up in the lung. So you can see here, blue is control, yellow is COPD, red is an IPF, and basically there's a loss of alveolar type 2 cells and alveolar type 1 cells, and an increase in ciliated cells. We also saw a dramatic increase in aerocytes and GCAPs, again suggesting damage to the alveolar exchange unit. Now this is important because when we thought about pulmonary fibrosis, people called it the disease of fibroproliferation and other things. There was no focus on the fact that actually most of our patients, the reason they die is because they lose diffusion capacity, not restriction. They use the ability to breathe. And the other thing that sort of showed up and was very impressive was sort of these collagen 15 positive cells, endothelial cells. Usually in the normal lung, there's no collagen 15 endothelial cells. They only show up in the central bronchi or close to the pleura. And in IPF lungs, you see a lot of them. You see here's a fibroblast foci, the epithelial cells, and here's the endothelial cells, the stain. And this is interesting because the lung has actually two sources of blood supply. The majority is pulmonary, and then you have the systemic vascular supply that supplies the bronchi and the pleura. And collagen 15 is typical of these, suggesting that we have basically a new population of cells that we were not aware of. What about other things? So fibrosis, of course, the disease of fibroblasts, and we saw some increase in cells that we called myofibroblasts, but the main cell that really characterized fibroblasts in the IPF lung were sort of cells that express CTHRC1, express also, they make a lot of collagen, and you see here, so this is colored by the disease is blue, is red, and control is blue. See, this is sort of the cutting edge of the disease, and you see here is CTHRC1. Every dot here is a cell, and you can see that this is sort of the cutting edge fibrosis. Now, these cells have been also first identified by Dean Shepard's group, and more recently sort of better characterized. Now, we wanted to know how do these cell forms, or what happens to fibroblasts. And the problem when we do human lung data, of course, you can't really do time course. You then can get biopsies. So we used a model generated by Jim Hogg and implemented by Van Herse and by Bart and by Wim Voets and Jim McDonough at Leuven, in collaboration with June Ding and Ziv by Joseph and Jonas Schupp and Zitinian and Faridari and Taylor, who does everything, in which we take differentially affect regions in the lung, we slices for them. And this is a normal lung. This is a normal lung in IPF patient. This is sort of intermediately involved lung, and this is sort of progressively. And we did nuclear-seq these lungs. We did 12 IPF lungs, 11 control, 3 regions. And we did identify all the cell type. And the question, could we identify fibroblast trajectories? So connecting to the previous talk, basically, we applied an approach that was developed by June Ding who's now at McGill. He was at Carnegie Mellon at the time. And basically, this is a deep neural network framework tailored for time series for single cell data. And you can pick up the preprint here. And basically what it does is you give it the cell data, and it sort of, after embedding, identify the cell populations and generates hypothesis about temporal dynamics and regulation, can identify perturbation and suggest way to reverse them. So we applied this data to mesenchymal cells in IPF. All samples were characterized in basically what we called 4 conditions, so stages: healthy, control, or IPF stage 0, mild IPF1, moderate IPF2, and severe IPF3. And this is sort of their the UMAPs generated by Yanagi. And what we can see, and hopefully you can see this because it doesn't project that amazingly, is that there is an increase with the stages in sort of what we call fibrotic fibroblasts and the CTHRC1 fibroblasts. And then we can identify different trajectories. So an example, you can see some cells, like this is lymphatic endothelium, they don't really change, whereas others have just a little bit of splits. But look at this, alveolar fibroblasts actually splits substantially during their phases, sort of suggesting that there's more of a diversity in their phenotypes. And we can, let's say we apply this, and we can actually identify the transcriptional regulatory events that determine every one of these events in every one of these tracks. And we can also identify dynamic markers, which are markers that change during the progression from one phase to the other. So in this case, I just picked collagen markers, and you can see that some collagen markers are increasing with disease severity, like collagen 15 and 1 and 3, and some are decreasing, like collagen C and other things. And what was cool is that we actually had decellulized tissues from these same exact areas that we extracted the cells. So we did decellulized matrix proteomics, and it actually confirmed the increase in these extracellular matrix and the decline. So really suggesting that the classification and projection of Unagi really works. The other thing that Unagi can is it can identify pathways and then suggest therapies that would reverse them. And this, we're only doing fibroblasts here. And it's interesting that some of the pathways were perturbed in fibroblasts are very, very obvious. TGF-beta signaling, actually, as you can see here, lung fibrosis shows up, really reassuring. ECM organization, collagen formation, collagens. But the other thing were things we did not necessarily expect to see: natrin signaling, syndecan signaling, and calcium signaling. And then here are sort of the perturbed ECM molecules. And basically blue is IPF fibroblasts and yellow is early. So you could see that there is a sort of a movement. And here are some drugs that are predicted to reverse the perturbations. So it's really was reassuring to see that nintedanib, FDA-approved drug, was actually predicted. But there were other drugs that were not known, and this is using public database. And we sort of nifedipine was sort of interesting to us. It's a sort of antihypertensive calcium entry blocker drug that's commonly used. So we took human precision cut lung slices, induced fibrosis using the fibrotic cocktail, and then treated them with the drugs. And you can see here that this is collagen in 1A1 and CTHRC1 in fibroblasts. And you can see the fibrotic cocktail sort of induces it, both in this and here. And then treatment with nifedipine or nintedanib or nifedipine really reduces the expression of both collagen 1A1 and CTHRC1, and also the sort of the number of cells. And what is cool is when you do this analysis and you use single cell analysis as an outcome in PCLS, we can now compare the global effect. So we could calculate, for instance, the distance of the experiments globally. And you see, this is sort of the distance between control and fibrosis. But actually, when we give nifedipine or nintedanib or both, we see that the distance from normal goes dramatically down, suggesting not only that we're measuring one outcome measure like collagen, alpha-muscle actin, but the gestalt of the tissue changes. And here also, what is interesting is these are the predictions, the in silico prediction of nifedipine effects on PCLS compared to the actual real-life prediction. This is on differentially expressed genes. This is on more extracellular matrix. And what's impressive is that basically all of the predictions of Unagi were confirmed. And so Unagi is sort of a machine learning. It's not a machine. It's actually a regenerative AI tool that uncovers new cellular trajectories in fibroblasts and IPF. Can identify trajectory-specific perturbations and therapeutic candidates. And we validated the data using proteomics and PCLS. The web tool will be released soon. Unagi was recently highlighted in Nature in a paper in a sort of a piece about tools for PCLS gene expression data. And it's also hopefully in a final revision in Nature Bioengineering. What about other cell types? So there's been a lot of said about the macrophages in IPF, and definitely if you're looking at impure cells, sort of what's called the SPP1 or spontaneous merticate profibrotic macrophages, which express also a lot of MMP9, a lot of kinases, definitely dominate, and they've been found in every data set and sort of originally published by Sasha Misharin. And I will not go into them in detail. The other cool thing was when we were analyzing the data was, and this is really the fun of doing omics, is that we found actually cell types that we never saw before. So we found a population of cells that were clearly epithelials, but we have never seen them before. And we call them aberrant basaloid cells. We call them basaloids because they express P63 and keratin 17, but not keratin 5. And they're definitely epithelial, but very unique. They express actually collagen 1A1, which is never expressed in epithelial cells. They express N-cadherin, which is never expressed in epithelial cells. Clearly, cells have epithelial mesenchymal transition feature. They have the machinery to activate TGF-beta. They express alpha-V beta-6. That is now an integrin that activates TGF-beta at the tissue level, and its inhibitors are studied. They express senescence markers, P21, P16, and GTF15. They also express MMP7, which is known, described by our group as probably the most validated for blood biomarker for outcome. And when we stain for them, and this was published in this original paper in Scientific Advances, they are sort of at the edge of fibroblast foci. So really exciting result. New cell type, or previously undescribed cells that have all the machinery for TGF-beta activation, senescence, EMT, MMP7. So really dismiss our knife of fibrosis. And then to try to understand where the cell comes from, what is their source, we actually have used the same thing, differentially affected regions of the IPF lung. And when we looked at the nuclear-seq, the clear thing was we saw all the cells that I told you, but we saw aberrant basaloid. We also saw a lot of intermediate alveolar epithelial cells we couldn't really easily characterize. And this is sort of the nuclear-seq data that you see the different cell types and the emergence of intermediate cells. And also you see that with progression of fibrosis in the human lung, you see the increase in aberrant basaloid cells and intermediate cell forms. This is sort of a UMAP. So here every dot is a cell basically, and we cluster only epithelial cells. And you can see alveolar type 1s are here, alveolar type 2s are here, aberrant real basal cells are here, aberrant basaloid cells are here. And there's this sort of bridge of cells that goes between alveolar type 2 cells and alveolar type 1 cells that potentially leads also into aberrant basaloid cells. And you can see that there's distinct markers like P63 is in basal cells and aberrant basaloid cells. CPA6 is only in aberrant basaloid cells, and this sort of marker, N-cadherin, the marker for EMT, is really only in the aberrant basaloid cells. And CTSE looks to be a marker of these bridge cells. So we said, can we actually use this data to really understand what happens early or late? And this is sort of a summary of a lot of work that was done by Taylor Adams, basically applying force-directed PAGA and seeing what are the genes that sort of drive toward the aberrant basaloid cells. And you can see that it looks like that the majority of cells are coming from alveolar type 2 cells, but there may be also some alveolar type 1 cells, and I call it the drinking bird model of epithelial cell plasticity in IPF. And this allowed us actually to identify markers for early and late cells and the bridge cells. So surfactancy definitely goes down. And then there's the emergence of CTSE that is a marker of intermediate cells, CPA6 and GDF15, sort of a progressive cells, and then we get P63 with the full definition of the aberrant basaloid cells. This allowed us also to do stains, and by staining multiple combination of surfactant and cathepsin A and surfactant and CPA6, we were able to show that in the IPF lung, never in the normal lung, we actually find these intermediate cells that express mixtures of different marker. Can this be modeled experimentally? So Herb Schiller has shown it in precision cut lung slices. Again, a piece of lung that we cut, inflate with low melt agarose, cut it using intervibradome and then culture. So we did a time course analysis looking at 0, 12, 24 hours, 48 hours, 72 hours, 96, and 120, and treated, we did controls and fibrotic cocktail. And what we saw was one thing. One is after the culturing of the PCLS, these tissues actually has undergone a dramatic trauma by being cut and then put in media. There's a dramatic boost of proliferation, which you can see this is untreated cells, but this is actually suppressed by the fibrotic cocktail. And we see the emergence of actually aberrant basaloid cells. And here we see sort of the different cell type, and you can appreciate how, remember in the previous UMAPs, we just have type 2s, type 1s, a little bit aberrant basal cells, and a few intermediate cells. Here, it's sort of the picture is really dominated by multiple intermediate forms, and you see the different names that we gave them of 81s and 82s, and the emergence of aberrant basaloid cells. And when we compare these aberrant cells to IPF lungs, we see there's a lot of features that are similar. Aberrant basaloid cells emerge from day 3. There's multiple intermediate form, and the fibrotic cocktail treated lungs actually show cells that really mimic the IPF cells, suggesting that we could use them to studies. So how different are these aberrant basaloid cells from regular cells? Are they really a different cell type? So we used ATAC-seq and the multi-ohm system, using both ATAC-seq and single nuclear RNA-seq to look at that. And what we see is when we did it, we saw that the nuclear-seq basically replicated the same data structure with 82s, 81s, intermediate cells, and aberrant basaloid cells. And when the cool thing is that the chromatin accessibility map also replicated the same structure. And you see aberrant basaloid cells, you see intermediate cells, you see type 2 and type 1, so very similar. And then when there's a sort of the markers of the different cell types, and you can see that the markers of in this slide, you see RNA on top, chromatin expression below, and chromatin accessibility below. You see these are the markers, so they're always, the chromatin is open. And here's for aberrant basaloid cells, you see sort of the typical markers are all chromatin accessible. And what's impressive is that these cells have way more accessible chromatin than any other cell in the lung. And you can see sort of the transcription factor binding sites that are identified. And what's common to all of them is sort of the AP1 site. And you can see here when we look at AP1 and just look at the peaks for all cell types, so they're a little bit different, right? But look at the aberrant basaloid cells. They include a little bit of this, but actually really go all over the place, suggesting that really they're at very different cell state. And that's been shown, the sort of this AP1 signature is very typical of senescent cells. But maybe this is a less phenomenon. All of our lungs are basically end stage. So we were lucky to have a collaboration with Venerino Politi and Claudia Ravaglia and Sarah from Florence, and they provided us with biopsies of patients with early pulmonary fibrosis. Many of these patients have interstitial lung anomalies. And initially, we just wanted to prove, so this is sort of a cryobiopsy, and we did both spatial transcriptomics and nuclear sequencing, and we were able to find aberrant basaloid cells. We were able to find all the features of the IPF lung, especially the loss of 81 and 92s, and the HRC1 fibroblasts, not necessarily all the other things. And you can see here when we're looking at blue is control, yellow is early IPF, red is end-stage IPF, green is other ILDs. You see that the early disease, there's a substantial loss of alveolar type 2s, and also the emergence of these intermediate cells, suggesting that this is an early phenomenon. And we can also find the aberrant basaloid cells. But what I think is interesting is that you see that you have sort of no intermediate cells in normal, increase in early, and a little bit of a decrease in IPF. And then aberrant basaloid cells start to increase. You can find them in the normal lungs, start to increase in the early, and then show up in full force in the end-stage lungs. And Oriane Justet did this analysis and showed that the emergence of aberrant basaloid cells associated with progressive disease in these patients, and we're now extending these observations to a larger cohort of 125 LA samples of them, 50 IPF. And we're going to do snRNA-seq and spatial lung, and we have also the blood and the BLs. So what is actually IPF? In the last sort of 4 minutes, the model that we propose is that we have sort of loss of alveolar type 1s, and then alveolar type 2s respond to injury. They want to become alveolar type 1s, but if they have short telomeres or they're genetically predisposed, they basically fail. And although they really want to be alveolar type 1, some of them die, some of them get stuck in intermediate form, and some of them senesce into aberrant basaloid cells that then drive ER stress and release of dumps and activation of TGF-beta. And basically whole hell breaks loose. The cool thing, of course, is that now we could sort of identify theories. So the features of IPF, as we describe it now, is loss of respiratory units, proximalization of the distal lung, shift in archetypes of alveolar fibroblasts, recruitment of monocyte-derived fibroblast macrophages, progenitor failure with stunted differentiation, and deluge of intermediate form. That's probably the key element, and the emergence of aberrant basaloid cells, a distinct cell type with an epigenetic senescence signature. And then, of course, there's a potential for intervention in a cell-specific biomarker. Now, I want to go back to sort of thinking about pulmonary fibrosis. So when we're thinking about tissue, we usually think about tissue homeostasis. You have regulated variable, a sensor, homeostatic signal, and an effector. So this is sort of, when the effector fails, there's going to be damage control. So could our cells fit in? And I want to go back to one of my first slides that I discussed the lung, the primary cell, and sort of the support cells. So when you want to think about this, one thing that is obvious, most of the cells in the lung don't do a lot. Alveolar macrophages, alveolar fibroblasts in normal state have not a lot of work. The majority of mitochondria and activity is one cell, which is the alveolar type 2 cell. The alveolar type 2 cell is a factory for surfactant, regulates fluids absorption, so it has a lot of ATP requiring channel, and serves as a local progenitor for ATE1s. And this image, which was generated by Maurizio Ciaccoli, shows also that the alveolar type 2 even moves when there is injury. And there is the highest number of mitochondria. This basically this, and we know that it is connected to IPF. And I think there's a clear system, which we all know, which is a clear principle. A system will most likely fail at its most stress component. And I think the alveolar type 2 cell is probably the failed cell in pulmonary fibrosis. So now we throw in the cells. We have the regulated variable, which is probably the gas exchange unit, a sensor, which I don't know what it is, and which is a type 2 cell. And if the type 2 cell fails to differentiate and it becomes an aberrant basaloid cell, this actually drives multiple processes, leads to aberrant repair, into this recurring model of fibrosis. So now we can explain the progressive feature and all the tissue features of the disease. And of course, we'll need way more work to actually validate this model. So I'll stop here and take any question. This is sort of many of the members in the lab. We have now updated photos every end of year. I invite people to my home, cook for them. This is a lot of fun for me. And I want to stop here and thank you for all for your listening and for inviting me, and glad to take any questions.
Thank you for this amazing talk. In the interest of time, we will move on and have the question in the Q&A in the end. Please go along and ask a question in the private chat. And if you can stop sharing, then I will present our final speaker. So that will be Dr. Federica Genovese, the Director of Translational Science. With over 15 years of experience focused on extracellular matrix, Dr. Genovese has developed and applied biomarkers to both clinical and preclinical studies supporting drug development efforts. Her extensive expertise in extracellular matrix and biomarker research has led to numerous publications, contributing significantly to the field. In her presentation, utilizing translational biomarkers for drug development in fibrotic lung diseases, Dr. Genovese will discuss how biomarkers can accelerate the drug development process by providing crucial insight into disease progression and treatment efficacy. Please welcome Dr. Genovese.
Thank you very much, Sarah. So it's my pleasure to finish this interesting session by talking to you about biomarkers that are applicable in the translational space. Let me start by saying that when we talk about biomarkers, we would of course like to have biomarkers that are diagnostic, that can differentiate patients with a disease. But even more importantly, we would like to have prognostic biomarker, which can identify patients that progress in a disease. And of course, the moment that we have treatments available for a class of patients, we would like that the biomarkers change with the treatment. So we can actually use them across all different needs in the clinical space and also with the within drug development. If you are here, if you are connected, I think that you share our vision that extracellular matrix is very important and at the center in many pathologies. You can see here all the different disease areas that we believe extracellular matrix is very relevant in. At Nordic Bioscience, we are very focused on ECM, and we published books around it. We participate strongly to the Extracellular Matrix Pharmacology Congress, which also sponsored this symposium. So thank you for being part of the community. And I would like to do a small introduction to our technology, because the previous speakers didn't explain how the biomarkers are designed. And in case you're new to what we do at Nordic Bioscience, maybe you require a little bit of introduction. So first of all, we like to say that collagen is not just collagen, and collagens are the main proteins in the extracellular matrix. And as you can see from this drawing, they have there are 28 types, first of all, and then they have very different function, structure, and localization. And we aim at quantifying the remodeling of all these molecules in understanding better when we find fragments from this molecule in circulation or in urine, what does that mean? Which kind of process are they actually describing? We are also very interested in looking at the cryptic signals of the matrix. So looking at fragments that when they are released by means of fragmentation from the mother molecule, they acquire bioactive potential and they actually signal to cells. And one of the biomarkers that I'm going to talk to you today is called endotrophin, and it's actually one of those signaling fragments coming from collagen type 6. So we want to be able to distinguish between the processes of formation of a new collagen and degradation. These are very different processes that reflect different triggers that can happen in the tissue and can trigger then the progression of the disease. So when fibroblasts produce collagens, what happens is that when collagens exit the cells, they release propeptides. And these propeptides can be detected in circulation or in urine through specific antibodies that bind the cleavage site of the propeptide. When we find a propeptide circulating, we know that a new molecule of collagen has been formed. So these markers can be used as proxy for collagen formation. Once the propeptides are cleaved off, collagens can assemble into fibrils and then into fibers, can accumulate in the tissue, and then can be subject to degradation by proteases. So when proteins are degraded, they form these small fragments, And we target these small fragments with specific antibodies that target the cleavage site. So this way we detect an active process of inflammatory-driven degradation of the matrix by proteases. So this is the way that we distinguish between the different processes. So in the same molecule, we can actually have several different fragments, which we identify with different assays. In this talk, I'm going to focus mainly on two biomarkers of fibroblast activity, which are both biomarkers that target the propeptides of collagen type 3 and collagen type 6. So here is the propeptide of collagen type 3 detected by the assay called PROC3, which binds at the cleavage site of the N-terminal propeptide. And we also have an assay which we call PROC6, which targets the C-terminal propeptide of collagen type 6, which is one of those bioactive molecules that I was mentioning before called endotrophin. Endotrophin has been associated to several deleterious processes, such as pro-inflammatory and pro-fibrotic effect. So the detection of this fragment in circulation can also reflect the release of this dangerous molecule, and therefore the active role of this molecule in disease progression. So these are the two biomarkers that I'm going to talk to you about, and that's why they deserve a specific introduction. So in my talk, I'm going to talk mostly about IPF, idiopathic pulmonary fibrosis. I'm going to show you a couple of slides on clinical data, and then I'm going to show you how we can apply the same biomarkers translationally in different models. So in IPF, what we notice is that both fibrogenesis described by the biomarkers PROC3 and PROC6, so biomarkers that detect formation of collagen type 3 and 6, and therefore fibroblast activity. And also biomarkers of tissue destruction. Here we have, for instance, a biomarker for collagen type 1, collagen type 3, and collagen type 6 degradation, mediated by matrix metalloproteinases. Both processes are important in IPF. You can see here that the levels in IPF patients compared to the healthy, which is the dotted line, are much more elevated for all biomarkers. So these biomarkers are diagnostic, but most importantly, they are prognostic. They are even more elevated in patients that progress compared to patients that do not progress. And this is true for all of these biomarkers. When patients with IPF have elevation in circulation of several biomarkers, then we can see that these are the patients that fare the worst. So there is a direct association with the number of elevated biomarkers and the lowering in forced vital capacity and in the oxygen level, the LCO levels. So there is a direct association with severity of the disease and number of elevated biomarkers. And the more biomarkers the patients have elevated, the higher the risk of mortality. So you can see here that these are really biomarkers that can describe the active process of remodeling in the lungs that are associated with the progression of the disease. The good news is that this biomarker can be modulated by treatment. So there are actually treatments that work in the IPF field, and we see a decrease in these biomarkers when the treatment is applied to patients. So here is an example with a dual integrin inhibitor, bexotegrast, in which we see that there is dose-dependent decrease in the levels of PROC3, so fibroblast activity biomarker in serum, and that the treatment is efficacious in favoring or in lowering the portion of decliner in FVC. We also saw that both PROC3 and PROC6 were decreased by a JAK inhibitor significantly in a dose-dependent manner. So again, another biomarker that has an effect, a direct effect on fibroblast activation. And we also have this study, which was a phase 1b with the omeprazib, in which the biomarkers PROC3 and PROC6 were reduced only 10 days after treatment. So these markers are highly dynamic, and they can be reduced in presence of treatments that have a direct effect on fibroblast and on the pathways that activate fibroblasts we can see that immediately in the circulating biomarkers. And that's, of course, a big advantage because we can see the effect of treatment very fast and in a non-invasive way. So now that I gave you a very quick overview of what these two biomarkers can do in clinical samples, let me guide you through preclinical models in which we see similar results. So the ideal scenario is that we have biomarkers that we can apply in the clinical phase, but that we have initially studied in preclinical model. So both cell models, tissue models, and animal models. Here you can see an example in which PRO-C6 was decreased by omeprazole, which I just showed you before, in scarina jar, which is a cell model, which I will describe you shortly, in a precision cut lung slices, and in the clinical trial that I just showed you. So we see actually an effect, a dose-dependent effect on this biomarker with this drug across different platforms. And it's very important to have biomarkers that can be applied translationally because you can actually already see an effect on the biomarker that you will use in clinical in very early stages of drug development. So let me start by talking to you about cell models. So the scarina jar model is a model that we have used for many years at Nordic Bioscience. It's a fibroblast monoculture model in which the fibroblasts are grown on a plate which contains FICOL, which is a crowding agent. We have this model developed for several different cells, such as lung fibroblasts, but also hepatic stellate cells and cardiac fibroblasts. And you can see here that you can appreciate the morphology of the cells and that the fact that they deposit collagen in the well. We usually run the model for 12 days, in which we treat the cells with different treatments every 4 days. We then assess the biomarkers that I showed you before in the supernatants. We can do a time course evaluation, or we can just measure at termination. And then we see the effect of the treatment that we test in this system on the fibroblasts, on the fibroblast ability to produce collagens. So we focus on using fibrotic cocktails, which are a group of cytokines that act on different pathways in the fibroblasts. And you can see here this cocktail that we are currently using, which we call the IPF fibrotic cocktail, because it's derived from a publication in which they saw which cytokines were present in the bulk fluid of IPF patients. So we reproduce this cocktail, and then we treat the cells with these compounds, and we observe the increase in the biomarker levels in the supernatants. So here you can see an example of one experiment that we did in which lung fibroblasts were treated with the fibrotic cocktail, and you can see here an increase, a large increase in the levels of collagen type 3 formation, collagen type 6 formation, and fibronectin formation. Treating with nintedanib, which is one of the approved drugs for IPF, we can see a clear antifibrotic effect of this drug that is dose-dependent in decreasing the levels of all biomarkers in the supernatant. You can also appreciate here the lowering of the levels of collagens with a serious red staining with treatment. We saw the same using another treatment, which is a prostacyclin analog from United Therapeutics. And this treatment is actually in a phase 3 study in IPF. We can see that there is also a very nice inhibition in all biomarkers evaluated with no effect on the viability of the cells, so on how many cells are alive or dead. You can also see in the levels of collagens in serious red staining that the treprostinil has actually a very good effect on that. So we recently ventured into developing a more advanced model for a monoculture of fibroblasts, which is a 3D model, which we call 3D Profib. Instead of culturing cells in plastic wells, we now culture them in specific plates which have at their which are precasted with a PEG hydrogel, which has a gradient of stiffness. So the cells actually can enter the gel, can penetrate the gel, and then they can form the networks between the cells, and they can grow in a true 3D fashion. You can see here, you can appreciate the difference in the cell morphology compared to the 2D, in which you can see here these elongated cells that form networks with each other, as well as in the formation of this lattice of collagen in the well. The way that we run the model is similar to SCAR in a jar, in the sense that we run it also for 12 days. We have an initial growth phase in which the cells penetrate the gel, and then we start stimulation. We measure the biomarkers in the supernatant like we do for a SCAR in a jar. We have initiated the development of this model in cardiac fibroblasts, and this is the reason why I'm going to show you some data on cardiac, but we now also have the model for lung, and I will also show you data on that. So what we observed in cardiac fibroblasts is that when we grow the cells in 3D here on the right, and in a normal setup of SCAR in a jar, we see a very different expression of the collagens with the stimulation of different cytokines. In general, it seems that the cells grown in 3D are more sensitive to PDGF and TGF-beta, depending on which marker we look at, compared to cells grown in 2D, which are only sensitive to TGF-beta. You can also appreciate that in 2D, here we could only measure collagen type 1 and collagen type 3 elevation from the untreated culture, while in the 3D, we see an elevation in all markers evaluated. So collagen type 1, 3, fibronectin, collagen type 6, and even collagen type 4, which is historically thought to be produced not by fibroblasts, but more by endothelial and epithelial cells. So there is a clear difference in the production of collagens when cells are grown on in different environments. And when we stimulate the cells, you can see here, you can appreciate that the cells change morphology. They proliferate when they are stimulated by PDGF. They differentiate when they are stimulated by TGF-beta. And then they produce a lot more collagen than when they are not stimulated. We can also appreciate that they produce different collagens according to different stimulation. It seems that PDGF mostly promote the growth of collagen type 4, fibronectin, and collagen type 6, while both PDGF and TGF-beta stimulate the growth of collagen type 1 and 3. Keep in mind, these are different collagens. Collagen type 1 and 3 are interstitial collagens, while collagen type 4 is a basement membrane. Fibronectin is the first extracellular matrix that is produced. Collagen type 6 is also in the interface between basement membrane and interstitial matrix. They may be collagens that help the cells move, and therefore a proliferating cytokine will induce first the production of these collagens and this extracellular matrix, so that it can help the cell to bind to the matrix and proliferate. So it is a model that allows to investigate these different aspects of cell biology. But most importantly, what we are interested in looking at is if we can use this model as a drug development tool. And so also in this case, we treated the cells with a drug that affects fibrosis, is again omeprazole, which I showed you before in the clinical data. And we can see here a dose-dependent reduction in PRO-C3 and PRO-C6 with after stimulation with either PDGF or TGF-beta. So this system is suitable for the drug screening, and we have now developed that also for lung. So you can see here how the lung fibroblasts appear during the days of growth, and that they are more and more forming network, and cells are mostly alive. And you can see here a close-up of the cell. I can see you, Sarah.
Yeah, so just in the interest of time, we need to stop in 3 minutes.
Okay.
So we have some time for Q&A because there comes a lot of questions in.
Okay. So I just show you this data on nintedanib. As I showed you before, nintedanib decreased the levels of PRO-C3 and PRO-C6 in 2D, and it does so as well in the 3D model. And to go a bit quick in 3 minutes on the other 2 models that I want to show you, the fibrotic cut lung slices is an ex vivo model that is used pieces of lung that are cultured, and that they are also used as a mean to test drugs. Here you can see again omeprazole being able to reduce the levels of PRO-C3 and PRO-C6 in the supernatant of precision cut lung slices. And other drugs like this interleukin 11 receptor blocker and again nintedanib and omeprazole also decrease the levels of PRO-C6 in this model. And I was kicked out. And my last, if I can present, okay. The last thing is bleomycin. Bleomycin is the model that is the most used in the IPF field. And here we see an elevation of PRO-C6 and PRO-C3 in the bleomycin model compared to the saline. That corresponds also to a lot of fibrosis in the bleomycin lung. And we see a reduction in the level of PRO-C3 with treatment with nintedanib also in this model. So again, the same results, you can see them from the cell model to the tissue model, precision cut lung slices, to the animal model, the bleomycin model. So key takeaway is that fibroblast activity is very important in IPF, especially PRO-C3 and PRO-C6 are the main biomarkers we look at, and they're modulated by treatment. And we can use the same biomarkers translationally. And my last slide is to thank the people that actually did the work. The translational team, especially Mark, Maya, and Stine, that did all the Skari-Najjar and 3D work, and the respiratory team, especially Jenny, Diana, Filippa, Helena, and Sarah, that produced all the clinical data that I showed you today. So thanks a lot, and now we can open for questions.
Thank you, Federica, for wrapping up so quickly. So if I can ask Naftali and Lina on the stage, and then I think we have some questions. So I got one from you, Naftali. Which type of macrophage is most involved in lung fibrosis? And the biomarkers could help for identifying all macrophages.
So what we think is common to most of the things in pulmonary fibrosis is actually monocyte-derived alveolar macrophages. The actual real alveolar macrophages are not that changed. And these cells have a sort of a stereotypic signature of genes, including osteopontin, MMP9, CHI3LK1, so YKL40, KIT1, MERTK. There's actually we have a paper in Science Translational Medicine with a group from Pittsburgh that they discovered a PIT marker, C1K1, that probably hits a population of these macrophages.
Okay. And then another question is, do you know if a clinical compound have demonstrated a modulation of apparent basaloid cells in IPF clinical trials?
Well, we don't know if it affects in clinical trials, right? Because there's no way to measure it. Although in theory, if you had a clinical trial that reduced, I don't know, MMP7, let's say, probably that would mean that it's reducing this abnormality, but it's hard to say. What we know from our PCLS that's unpublished is that most compounds we tried change the behavior of these cells, but it does not change their quantity. And I think, but that also makes sense because many of the compounds we have were designed to treat fibroblasts. So so we're working on ways to try to reprogram these cells.
Yeah, so you think it could be a drug target?
Well, actually, there is one drug target, the integrin inhibitors, alpha V beta 6. Definitely, alpha V beta 6 is expressed in orbital type 2 cells, a lot in basal cells, and a lot in aberrant basaloid cells. So this could be a potential target.
Okay. Very interesting. And then I have one for you as well, Line. Can you explain a bit more about the data you used and how it was used in clustering example? Was it a prediction you ended up with?
Thank you for the question. So I feel like if Federica had been the first one to speak, I think more people would understand what goes in, and it is these specific protein fragments we measure in the blood, and we get that measurement out and we implement it into this prediction model. And when I say prediction model, it is not necessarily because we make a prediction. It's just the wording you use in machine learning. It is more these prognostic endotypes you find. So it's correct. It's not a prediction. It's a prognostic endotype we find.
And how do you usually need? How many biomarkers do you usually need to make endotyping work? Is one enough?
Well, I wish I could say yes, one is enough, but I simply don't believe it, and not after I saw Naftali's very, very great presentation where they showed these single cell experiment that shows there's so much underneath a biological questions. So I don't think there's just one biomarker. Hopefully we can boil it down so we only look at the primary biomarkers and can leave out some of the supplementary ones, but I don't think one biomarker is enough. And I would really like your opinion on this as well, Naftali, because now you're working with this huge single cell models. Do you think one is enough?
I don't know, to be honest. I'll say that it's not enough and then it will be enough, or the other way around, right? I do think that it would be, the way you're thinking about endotypes and combinations of markers is the correct way. I think that, like, maybe some patients are more proteolytic and some are more ECM secretors. We don't even know it yet, right? So ideally, groups like Nordic would get samples from thousands of patients and we could do it, right? And then the other thing is actually some patients are more like IL4 or TNF type of patients, right? So I think we'll have to have endotypes. And I doubt that now, and it's interesting, if you look at the prolific biomarker set, I think 6 biomarkers are out of what did they pick 13? I don't remember. But out of the biomarkers, 6 carry a lot of information. The others don't add much. But this also could be because of the patients we tested them. Right? So we have a tendency in the IPF community to overclassify. So I think sometimes the problem that the physician deals in the community is very different than the pure patient we pick for a clinical trial. So I think we may need to spread our wings a little bit wider. But again, hard to say. Yeah.
I think there come one more question for you. Naftali, would a pulse of depletion of apparent bathylated cells make space for regeneration?
No. So I think we do need to separate between probably IPF and other fibrotic lung diseases. I think in some of the other fibrotic lung diseases, the defect is going to be potentially fibroblasts or macrophage activation or this. I think in IPF, and it is sort of this progenitor cell failure, so type 2. So in theory, okay, I think if you slow down, if you get rid of aberrant basaloid cells, you definitely stop disease progression. To get the patients better, you need to find a way to encourage the growth of more alveolar type 1s or type 2s. Now, it could be that just by changing matrix, reducing stiffness, reducing ROS, you're going to improve their survival. But without targeting sort of the primary loss of the gas exchange unit, it's going to be very hard to improve this. I think we have now probably 10 or 12 strategies that will either slow down the disease or stop it completely. I don't think we have good strategies to make the patient better.
Sure. So, Frederica, I also have some question coming in for you. So, a quick one. Do you treat fibroblasts in starving media?
Yeah, we use a 0.4% FBS.
And do you know which range of stiffness of gels are used in the 3D profib model? And how were those values selected?
Yeah, I mean, I don't remember by heart the kilopascal, but it's soft. It's a very soft gel. This is based on what is possible mechanically with the pegs. We try different stiffness, and we see that slightly harder gels make the cells grow better, but we are talking still very soft material. Yeah.
And how did you come up with this profibrotic cocktail?
This was from a paper in which the bulk fluid of IPF patients was analyzed for cytokines. So this is the cytokines that were found in the patient's fluid. So we think these are probably relevant to instigate the disease, and that's why we replicate it in the cell model.
And then I have a more biomarker-related question. So what is the difference between PRO-C3 and PRO-C6?
It is two different collagens, of course. I think that the main difference is that PRO-C3 is the main, one of the main collagens in the interstitial space. So it's really the collagen that is responsible for the fibrotic scar. PRO-C6 is more a supporting collagen, and I believe that the reason why PRO-C6 is such a good biomarker is because of this bioactive role of the propeptide. So that's the difference between the two. We also put in clinical data in other diseases that PRO-C3 is more related to fibrogenesis, PRO-C6 with risk of outcome.
Thank you. So we are running out of time, but I think we have time for one last question for you, Naftali. So you mentioned that loss of endothelial capillary cells is an important change in IPF. Would pro-angiogenesis be an effective treatment strategy? And if yes, what is the limitation?
Probably not, right? Probably not, because unless I think you need to populate. You need to populate the alveoli with sort of the specialized aerocyte cells that do gas exchange. So I doubt that pro-angiogenesis, if there's a good chance that actually inhibiting angiogenesis may have a decent effect on reducing shunt, but again, hard for me to advocate for it any way. I think we'll learn actually potentially with the effects of the PDE4 inhibitors. Potential there's some effect on vascular. The same thing with prostacyclin, some effect on fibrotic cells. The emergence of the collagen 15 cells, I think, is actually a marker of angiogenesis. I think what the lung is trying to do is it's building an airway. So you're getting fibroblasts, you're getting endothelial cells. You actually get smooth muscle cells. So promoting angiogenesis is probably not the good way. Maybe promoting differentiation of aerocytes, yeah.
Okay, then I think one last question for you, Frederica. What are the advantages of the 3D model compared to the normal 2D scanner gel?
The 3D gives a more in vivo-like model. Cells are actually growing not on plastic, which is not their real environment, and in a much softer environment, so they don't self-activate. Moreover, the model per se allows for other observation, like morphological observation, immunofluorescence, staining. So it's also more advanced in terms of what can offer in observations, not just the supernatant. Yeah.
Thank you. Then I just want to wrap up and thank you to our three speakers. It's some very nice presentation, and hope to see all of you to our next webinar. Thank you for tonight.
Thank
Bye.
you.
For the invitation. It was a pleasure. Thanks.
Bye.